Hello, biology buddies. I hope that you, your friends, and family are all safe and doing well. This is lesson 5 from Unit B1, Cell Biology, and the title is Microscopes, Microscopy, and Magnification. Some nice alliteration there. What are we going to do for this lesson? We're going to learn how to describe the parts of a light microscope. We're going to be able to explain how to prepare a specimen, put it on a slide, and then use a light microscope to observe the specimen. We're going to be able to compare light microscopes and electron microscopes. And last but not least, we're going to learn what the magnification equation is and how to use it. You're going to need to remember the stuff from the previous lesson, lesson 4, on the SI units, specifically the prefixes, so millimeter, nanometer, micrometer, etc. There's not that many equations you need to learn in biology. So for example, in physics, there's way more that you need to remember, but the magnification equation is absolutely one that you need to master. It's very likely to come up on paper 1. Let's get started. As you can see, we have a diagram on the right here of a typical light microscope that you'll have in a science classroom. Microscopes are very basically used to magnify specimens, to make them look larger. And they do that by a combination of the eyepiece and the objectives. Whenever you're carrying a microscope, you want to have one hand on the arm, sometimes people refer to it as the neck, and the other on the base. These aren't cheap pieces of equipment. You want to make sure you carry it properly. Once you've put it down, you've plugged it in, you'll notice that the light source is here. There will be a button on the microscope to turn the light on. What are the pieces we need to talk about on an exam? First thing is you're going to look down the eyepiece, and the eyepiece actually already magnifies things about 10 times typically, so it's not just the objectives that magnify things. You'll then also have these bits here, which are called the objectives. Typically, in a science lab, you'll have a 4, 10, and 40 times magnification. We need to remember that the eyepiece is already 10 times. So if I'm looking using the 4 times objective, I'm actually magnifying the image 40 times. On the right-hand side, you will have 2 dials. You have the coarse adjustment, sometimes referred to as the coarse focus, and the fine adjustment. What they do is they actually move the stage, which is where you're putting your slide, up and down. So you're going to use those to focus on the specimen to make it clearer so that you're able to see it. So when people are talking about focus, when you're using a microscope, you are turning the coarse adjustment until you can see it, and then using the fine adjustment to get it as sharp an image as you possibly can. You always start with the lowest objective. Use the coarse adjustment, use the fine adjustment, make sure that what you're looking at is right in the center of the view, and then switch to the next objective. So from 4 to 10, you turn the revolving nose piece there, and then you repeat the process, coarse adjustment, fine adjustment. I know that sounds super obvious, but there's actually been 3 4-mark exam questions in the past that were literally, how would you observe a specimen using a light microscope? And people lost tons of marks because they didn't say you start with the lowest objective, and then use the coarse adjustment, fine adjustment, then move to the next one, etc., etc. So using a microscope, like we said, you place the slide on the stage. You have these things called stage clips that sit over top of the slide to hold it into place. You start with the lowest power objective, use the coarse adjustment, use the fine adjustment, and then move on to the next objective and repeat. Really simple. When you're using a microscope in lesson, you will often be asked to draw a diagram of what you see, and there have indeed been questions on an exam where they give you a picture from down a microscope and ask you to draw and label, so to do a biological diagram. A couple of really simple rules to follow here when you're doing your diagrams, but also if you get it on an exam. Number 1, which is the most important, only draw what you see, not what you think you're supposed to see. If you're an expert on cells, you've gone through lesson 1 a bunch of times, you know about the mitochondria, you know about the ribosomes, etc., etc. You're going to be tempted to add those because you think, oh, I've just messed up my slide. They must be there. I know they're supposed to be there, so I'm going to draw them. Don't do that. Only draw what you see. A light microscope does not have a very powerful magnification or even resolution, so you're not going to be able to see everything that you think you will. For example, if we were going to do the epidermis of an onion, so just some onion that you've peeled off, really thin layer, and put down on a slide and then stained, you will see something similar to this. And I know that my artistic abilities are second to none. Don't strain yourself trying to draw perfect rectangles like that. You're going to have that. You're going to see a circle, which is the nucleus, and then that's it. You're not going to see the cell membrane. You're not going to see the mitochondria. You're not going to see ribosomes. This is all you're going to see. You don't shade. You don't use color. You're not doing depth. You're not doing shadows. You're not going for tone or emotion. You're just drawing what you see as clearly and simply as possible. You want to label your diagrams. So on our diagram here, we're going to label the nucleus. Going to label the cell wall, and there's the cytoplasm. I'm not labeling anything else because I cannot see it. You'll notice I haven't crossed lines. You don't want to end up in a situation where your lines are going like this. That's a big no-no. It makes it more difficult to see. Nice straight lines that don't cross at all. Last but not least, you want to title your diagram and include the magnification you used. Remember, for the magnification, you're going to need the eyepiece and the objective. So multiply those two together. So for example, if I was using the 4 times objective, 10 times eyepiece, this would be 40 times magnification. So I'm going to label it as onion cells. You'll notice I'm putting the label at the bottom. And then I'm going to put 40 times magnification and underline that. Done, 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 done. Lovely. Sometimes when you're looking down the microscope, you will see a bunch of circles, perfect circles, lovely circles. Those are air bubbles. It means that you did not prepare the slide as well as you possibly could have. Even the best of us sometimes get some air bubbles, so don't beat yourselves up. But definitely don't draw those and start labeling them as cells. You're looking for plant cells. You're not looking for air bubbles. Next, we will look at how to prepare the cell for observation before you look at it. Actually, nope, that was a lie. One last thing. Don't draw the field of view, so that light circle that you see down the microscope. So you wouldn't want to have a diagram that looks like that. You don't draw the light circle, you just draw the stuff that you can see, not the circle that's created by the light coming in. You might also get a question asking you how to prepare a slide or prepare a specimen. Really, really simple. You want, again, if you're using it with an onion, peel as thin a layer as you can. They're going to be looking for that word thin. You want 1 cell thick because you're looking through it. So if you have multiple layers, it's either not going to allow the light through and you won't see anything, or it'll get confused because you'll be looking through a bunch of cells at once. You place it on the middle of the slide. You add the drop of stain or water to your specimen. For the stain, the reason you're doing that is to make structures visible. So to make the organelles, remember back from lesson 1, the word organelle, the structures that are in the cells, make the organelles visible. Different stains are going to, for lack of a better word, attach to different things. You don't need to know the names of the stains at all. It's most likely that you'll be using iodine for your onion slide. Then you put the cover slip at a 45-degree angle. That's this right here, with the edge of it in the stain or the water. The reason you're doing that is you want it to come into contact with the liquid, because then when you drop it down, it's going to push all the air out as it falls down. If you just dropped it straight on, you're going to trap air bubbles in there, and then you're going to end up with that problem of when you look down the microscope seeing a whole bunch of little circles. And that's it. So again, not an uncommon exam question. How would you prepare a slide, or how would you prepare a specimen for looking at under a microscope? Those 3 really, really easy steps. Really thin layer, put it in the middle of the slide, add your stain, cover slip, which is your very thin piece of glass that goes over top at a 45-degree angle, drop it down to push out the air bubbles, and you are ready to go. We're going to get into some key words now. Magnification, very simple, how much bigger the image is than the actual object or specimen. Resolution is a trickier one. Resolution is the ability to distinguish between 2 points. And I've said here a higher resolution image is clearer. This does not mean the same thing as focus. So focus was moving the stage up and down with your coarse and fine dials on the side there. Resolution is something that's different. The best explanation that I've heard about resolution is imagine a digital photograph. Could be one on your phone, could be one on your computer. It can be a nice, very clear picture, but as you zoom into the picture, it will start to get more and more blurry until it's difficult to tell 2 points or 2 pixels apart from one another. Right? Keep zooming in, keep zooming in, keep zooming in. Eventually the image will become blurry. The higher the resolution, the more you can zoom in and still have that picture be sharp and clear. Okay? So we're going to talk about electron microscopes in a little bit. An electron microscope has a much higher resolution than a light microscope. That doesn't mean at low power that the picture is clearer. What it means is as you magnify in, you still get that super crisp, clean picture. It doesn't get fuzzy, it doesn't get blurry. Doesn't mean the same thing as focus. So resolution is the ability to distinguish between 2 points, and that becomes super important as you increase the magnification. You can test this for yourself on a digital photograph. Zoom in, zoom in, zoom in. Eventually it will become very blurry. The lower the resolution of the photograph, so you can typically see that in terms of how big the photograph actually is in terms of memory. The bigger it is, the more memory it takes, the higher the resolution, the more you can zoom in. A low resolution photograph, especially ones that were taken from digital cameras when they first came out, you could barely zoom in at all and you'd already start to see the pixels. Okay, enough of a trip down memory lane there. Magnification equation. Very, very, very simple. And if you worry about forgetting it, it is common sense. The magnification is the image size divided by the actual size. Right? If it's 40 times magnification, that means the image is going to be 40 times larger than the actual size. The way that they're going to trip you up on the exam is that you have to check the units. Particularly if it's like a 3-mark question, you go, how is this 3 marks? It's super easy. You've missed the fact that they're asking you to convert units as well. So you need to apply the skills that we learned last lesson, where we talked about the prefixes, how to go from millimeters to meters, how to go from micrometers to millimeters. You have to have that memorized. You have to have that mastered. Remember, as you go from bigger to smaller, you're going to multiply by 1,000. When you go from smaller to bigger, each step you're going to divide by 1,000. Let's have a look at some examples. Super easy one to begin with. If a student is using a microscope to view an object that is 1 millimeter wide and the image is 100 millimeters wide, what magnification is the image? Now, most of you are yelling at the screen right now saying 100 times, this is way too easy, and you're absolutely right, but let's go through the steps anyways. Step 1, check the units. I've got millimeters and I've got millimeters, so I'm okay. I don't need to do anything there. Step 2, write down the equation. Magnification is equal to image divided by actual size. Step 3, I'm going to plug in my numbers. So my image is 100, my actual is 1, so that's going to be equal to 100 millimeters divided by 1 millimeter. I'm writing the units there just so I can double check later on that I haven't made a mistake, is equal to 100 times magnification. If I can get that written. Okay, super simple. Let's try a slightly trickier one. A student is examining the stomata, that's the hole in the leaf. There's more of them on the bottom of the leaf than the top. It allows carbon dioxide to come into the leaf to be used for photosynthesis, oxygen to leave the leaf, which is a product of photosynthesis, but also when the stomata is open, it's going to lose some water. That comes up near the end of B2, super important, transpiration. Okay, back to magnification. Step 1, let's look at the units. I have micrometers and millimeters. Ooh, okay, so I'm going to have to do a conversion here. It'll probably be a 3-mark question. I've got the actual, I've got the image size, they're asking me for the magnification. So step one, let's convert. Let's go from millimeters to micrometers. So I remember if I'm going from millimeters to micrometers, I'm going from a larger thing to the smaller thing, the next step down. So I need to multiply by 1,000. If that doesn't ring any bells with you, go back and look at the previous lesson, please. So my 5 millimeters is actually going to be 5,000 micrometers. Are my units good now? Yes, they are. Okay, I'm ready to go. Next step, write down the equation. Magnification is equal to image divided by actual, which is equal to 5,000 divided by 50, which is 100 again. Now you would get some marks if you didn't convert there. You also would have said, well, hold on a second, my magnification is actually less than 1, because you would have 5 divided by 50. So if you end up with a magnification of 0.1, that's actually making the thing, it's not making a big difference at all. So I, should clue in there. Let's have a look at another example. Example number 3, a student has drawn a diagram of a cheek cell. The cheek cell diagram has a length of 24 millimeters. The image has been magnified 400 times. What is the actual length of the cheek cell? Give your answer in micrometers. So step 1, let's check our units. Micrometers, millimeters. I've got a problem there. I also have the issue that they gave us the magnification and they've given us the image length. We need to find the actual length of the cheek cell. So it's slightly different than it was before. Step 1, they want the answer in micrometers. They've specifically said that at the bottom of the question. They will have a line where you write it and then the symbol for micrometers just to make sure that you understand it. So let's do 24 millimeters into micrometers. We just did this. If you're going millimeters to micrometers, you're going to multiply by 1,000. So I have 24,000 micrometers. So my image is 24,000. My magnification is 400. Next step, write out the equation. Magnification is equal to image over actual. This time I want the actual. So to get that free, I'm going to multiply both sides by actual, and I'm going to divide both sides by magnification, and I end up with actual size is equal to image divided by magnification. I can then replace in my values. My image was 24,000 micrometers. And my magnification was 400 times. Notice that there's no unit there, so when I divide this, I'm going to get an answer in micrometers. 24,000 divided by 400. Can cross out the 0, 0, the 0, 0. I end up with 240 divided by 4, which is going to be 60 micrometers is my answer. Let's do another one. This one is the classic question that confuses students massively, and then when they see how to do it, they end up hitting themselves in the head and saying, oh my God, I can't believe I didn't do that on the exam. And it is one of the main reasons that on the front of your exam paper, it will say, for your equipment, you need a ruler. Make sure you bring a ruler to all of your science exams. It's not just to draw graphs. Sometimes you actually have to measure things. What do I mean by that? This question, which is of a mitochondrion, so where aerobic respiration happens in the cell, remember back from lesson 1 in this unit, what is the magnification of this image? And you say to yourself, all that I'm given is this scale. They've forgotten something. They haven't given me the actual size. They haven't given me the image size. What am I supposed to do here? Amazingly, for this question, you ignore the whole picture. All you're going to look at is that scale that's right there, because the scale is to scale, that's why it's called a scale, to the diagram. So if that length is 1 micrometer, then that length in the image is 1 micrometer, which means what you're going to do is actually put your ruler up to the scale. You do not need to measure the length of the mitochondrion. It's not asking you for the actual size, it's asking you for the magnification. So you're just going to go to your scale, and I know that it doesn't line up perfectly. It was such a pain to try and drop this in. I'm not that great at using PowerPoint. I know it doesn't line up perfectly, but we're going to pretend that it does, and we're going to say that it goes to 31 millimeters. So what do we know then? We know that our image size is 31 millimeters, and our actual size is 1 micrometer. Why do I know that that's my actual size? Because it told us that that is 1 micrometer. Okay, so image is 31 millimeters, actual size is 1 micrometer, and we've taken what looks like a really hard question and we've made it super simple. Step 1, what do we need to do? We need to check our units. We have millimeters, we have micrometers. We can't have that, so we're going to convert the millimeters to micrometers by multiplying by 1,000. So I have 31,000 micrometers. Then we write out the equation, magnification is equal to image divided by actual. I know it seems a pain. Please write out the equation. Please substitute in the numbers. If you have a brain fart on the day of the exam and the examiner can see you're working and can see that you just made a really silly mistake, they can give you what's known as ECF, error carried forward. They will still give you marks even if you end up with the wrong answer, if you show them how you got there. If you just write down an answer and you made a mistake, then you're out of luck, you get 0. Magnification is equal to image over actual. We swap in our numbers. 31,000 divided by 1 is a magnification of 31,000 times. And yes, that is a crazy high magnification. There is no way on earth that you would get that using a light microscope. This was generated using what's known as an electron microscope. There's a whole bunch of different kinds of electron microscopes. You don't need to worry about that at GCSE. At A level, you learn about transmission electron microscopes, scanning electron microscopes. GCSE, don't worry about it, just electron microscope. So if you're looking at that and saying it can't possibly be the right answer, 31,000 magnification, you can't get that with a microscope. You can, just not a light microscope. And that brings us to electron microscopes. So most of what we've done for this lesson was with light microscopes. Remember when we drew our diagram of our beautiful diagram, I should say, of the onion cells, all you could see was the cell wall, the nucleus, and the cytoplasm. You couldn't see the ribosomes, you couldn't see the cell membrane, you couldn't see the mitochondria, anything like that, nothing in the cell except for those things, because those are the only things that are visible using a light microscope. The other stuff is too small. So if you want to look at that stuff, if you want to generate micrographs, it's almost like a photograph using a microscope, you need to use an electron microscope. And as you can tell from the name, these are used by shooting electrons at things. Why are they useful? They have a much higher resolution. Remember, the ability to distinguish between 2 points, and a much higher magnification. If we go back to our example here, 31,000 times magnification. You're not getting that with a light microscope. So those are the 2 big advantages. Disadvantages, they are much more expensive. They are much bigger, and the organism has to be dead. Why does it have to be dead? Because you are putting it into a vacuum. You can't have any air molecules in there. They're going to mess around with your electrons. It needs to be in a vacuum, which means that your organism needs to be dead. You can look at living things using a light microscope. You don't want to leave them on there too long with the light on, because it'll evaporate all of the water that's on the slide and then the things will die, but you can watch them while they're alive for a little bit. So we have our positives, we have our negatives. We have positives, we have negatives. What might the next thing be then? evaluating light and electron microscopes. You can have a question on an exam asking you to compare them, giving you a situation, asking you the advantages and disadvantages of using them. We need to remember before we do anything else that the word evaluate means something very specific. It means advantages, it means disadvantages. You might think of this as benefits and risks. And then finally, you need to make a conclusion. So if it asks you to evaluate the use of a microscope for a particular thing, at the end, you're going to need to say which one is better. You can copy out this table and have a go at doing it yourself. I imagine that you will have no trouble with it whatsoever. And here is one I made earlier. Light microscope, advantage, cheaper, organism can be kept alive. Disadvantage, much lower magnification, much lower resolution. You are not going to see as much, and when things get really small, you're not going to see them at all. Electron microscope, advantage, higher magnification, higher resolution. Disadvantage, cost, and also the organism has to be dead because it is going to be in a vacuum. Okay, that takes us to the end, so let's have a look back at our outcomes. Describe the parts of a light microscope. You need to be able to say what the eyepiece is, what the objectives are, and what the coarse and fine focus adjustments are there for. And remember, the coarse and fine focus adjustment move the stage where the slide is up and down, and that allows you to put it into focus. So if you get a question that says you have an image that's out of focus, what should you do? You do not change the objective, you do not change the slide, you do not get replacement eyes, you move the stage up and down using the coarse and then the fine adjustment to get the image clearer. Explain how to prepare a slide and use the light microscope. We just talked about how to use the light microscope. Don't forget that you need to start with the lowest objective and then gradually move your way through the more powerful ones so you get a more magnified image. To prepare a slide, we need to remember that we are putting the stain on to make structures visible, and then we are dropping the cover slip at a 45-degree angle so that you don't get air bubbles. Comparing light microscopes and electron microscopes, cheap but low resolution, low magnification versus expensive, high magnification, high resolution, but also the organism needs to be dead. Last but not least, we looked at a bunch of examples of the magnification equation. I have set some work for you guys on forms. I appreciate any feedback either on the form or on the videos. Stay safe, and I will see you next lesson. Bye-bye.